Hello and welcome to the Brain and Transcranial Photobiomodulation Virtual Summit. I'm Dr. Joe Doduro. My guest today is Farzad Salehpour. He received his master's degree in medical physics from Tabriz University of Medical Science in Tabriz, Iran. He is on the board, editorial board of Photobiomodulation, Photomedicine and Laser Surgery, the official journal of the World Association for Laser Therapy, WALT, and the North American Association of Photobiomodulation Therapy, which is NALT, and the official journal of the World Federation of Laser Dentistry. He has over 30 scientific peer-reviewed publications and 2 book chapters. He is ranked number 8 in the world as an expert scientist in the field of brain and photobiomodulation therapy based on the PubMed indices. He's the author of the definitive narrative review published in 2017 called Brain Photobiomodulation. He's the senior author of the forthcoming textbook, Photomodulation for Brain, from the Institute of Physics, published by Morgan and Claypool. It's my sincere pleasure to introduce you to my friend and colleague, Farzad Salehpour. Thank you, Farzad, for your time.
Thank you. Thank you so much, Dr. Joe. Thanks for having me.
Well, you know that you are number 8 in the world. You're the expert, and you are the man who's really started, I think, a lot of this process. Would you tell me, how did you get started in this field, to dedicate this as your life's work, to publish so many articles and so many? How did you get started? What brought you to this place?
Okay. First of all, thank you so much for this great opportunity, and it's an honor to be here. Actually, photobiomodulation entered to my life since 2015 when I was working on my Master of Science thesis. So I was working on the PBM4, transcranial PBM4 depression animal model, and then we focused on some of the anxiety symptoms and depressed indexes in rat model. And then I got interest into the topic, like how light and laser can work, because actually my previous study was on the physics, atomic and molecular physics. So laser was my interest from the beginning. So in 2016, we published the paper in Laser Surgery and Medicine, and after a month that I worked on the review article, focusing on the literature review of the whole studies on the topics of the photobiomodulation for depression. And so in 2017, that was accepted. And then I focused on the work on some of the laboratory studies, like the focus on the precognitive effects and other aspects, like working on the sleep deprivation and so on. So until 2018, I was working on just the laboratory stuff, but just when I got some free time, I was thinking that maybe it's time to, start with the big project, like the reviewing of the whole literature in this field. I mean, the brain PBM. So I worked on the preclinical and clinical studies, the whole papers, around 200 or 250 the various papers. And after 6 months, I wrote, along with my colleagues and Mike Hamblin as a co-author, and we wrote one of the good papers in this field, like Brain PBM Therapy Narrative Review, which has been published in the Molecular Neurobiology in 2018. And after that, I just working on some of the clinical studies and so on and so on, and then get into the book project, as you mentioned.
So, you know, to let people know, you are a medical, your study was physics. And I always think it's always good to have a physicist in the house because he's able to, you know, understand the complexities of this, but you've also made it very easy for people to comprehend. You know, you've, as you said, you've read over 250, reviewed over 250 articles just to write one article. So you have the ability to distill this stuff down and make hard science pretty easy for people to digest. So I appreciate that. Let's take a look at your slides.
Okay. All right. So today I'm going to talk a little bit in details about what we call brain PBM in terms of the whole mechanistic things, the biophysical things, and the clinical, implications. So as you mentioned, as a medical physicist, we have a couple of, concerns in this therapeutic approach because we are working on the lasers and interaction with the biological tissues. So this is very important to interpret the interaction between light and neurons. So this is very important to evaluate the exact amount of dosage, how many photons can reach, and what is the interactions type. And so I thought maybe it's okay that I just focused on all of these topics during my slides and representation. So let's see what happened. Okay, in the part one, I'm going to just represent some of the fundamentals in this area, as we have the biophysical and neurobiological aspects of brain PBM. So as all of you know, the PBM promotes neuroprotection and neuronal repair, which is one of the main and essential aspects of the light that when interact with the neurons or other types of cells. And this is one of the promising neuromodulation techniques. This is the neuromodulation techniques. So this is not kind of diagnostic methods using the near-infrared light, or even this is not some of the thermal therapy techniques. This is just the using specific dosage of light to stimulate or modulation of the neurons. So this is safe method, simple, pain-free in terms of transcranial approach, and intranasal also, and also inexpensive and easy to administer. So will be well tolerated by all patients almost in all clinical trials. So as I mentioned, this method is different from other types of light application in medicine, like transcranial optical imaging, optogenetics, photodynamic inactivation of neurons, and most importantly, laser thermal therapy, and most importantly, even neural photostimulation. Sometimes people get confused with the photobiomodulation with optogenetics and neural photostimulation. And we have to know that the output power in the photobiomodulation is the index, and the indication for what is the therapeutic method is the output power and irradiance range. And these two can show that we just use light to neuromodulation, or we use just light to stimulate the neurons, which photostimulation means. Okay.
So on this slide, just to let me know, so you're pointing out that output range varies, the irradiance range varies, and the fluency range varies. And you can use your little, you can use your mouse to kind of point us around a little bit, because this is called, you're using this as high level laser therapy, medium level laser therapy, and low level laser therapy. Those are the things that you're talking about. So the photon
Yes, sir.
intensity is determined by the output power range, the irradiance range, and the fluency range, right? That's what you're trying to
Yes,
point out.
yes, yes. Because in the optogenetic therapy or optogenetic methods, the scientists just use some of the exogenous materials and substances and then inject it into the neurons, and then we will have some of the irradiations like the blue light, or depends on the opsins, wavelengths. So that's not the photobiomodulation. Photobiomodulation, we don't have any kind of, injection of the extra substances. And so even in the neural photostimulation, um, photostimulation methods is different in terms of the pulsing, and also the mechanism, because in that method the mechanism is based on some of the action potential and ion channels.
Very good.
That and also the wavelengths is different. Longer wavelengths, more than 2000 nanometer are used in that method. So, yeah, photobiomodulation has specific, parameters. We have to take into account. So for delivering light into the brain, you can, as you can see in the figure, there are the various types of delivery approach that you can see. Transcranial is one of the most studied and applied method, using contact or non-contact manner, and also intracranial is another method that has been used during over the past 5 years, mostly on the animal, the rodents, and a couple of, studies on the primates. Okay, and intranasal photobiomodulation is another option which has been investigated in the clinical studies recently. From the 2017, we can see there's some of the clinical application in dementia, healthy, healthy subjects. Okay.
So there's also, Farzad, there's also the point of, this is transcranial using probes, which is single spot. And then we have companies that are using multiple accesses. And then we have companies or devices that are using pancranial devices. So this is a very good, different approach, but there's probably at least two more approaches that I'm sure that you're going to touch on as we go forward. Just to let you know. Beautiful. Keep going.
Yes, absolutely. Okay. And some of the other types and rare types of irradiation and approach, like the oral cavity route or even ear canal, and also have been, put forward in the literature as a potential, approaches for getting, for delivering enough light into the deep brain. And so, in terms of the intranasal, this is our the recent paper that has been published in the reviews in the Neurosciences journal, and we, how can I say, there are a couple of papers that photobiomodulation via the nostril-based applicators can improve well-being and mostly brain function. It can benefit brain. So, but in terms of some of the hypothetical approaches and hypothetical, how can I say, the methods, there have been some of the patents in the literature that have mentioned that maybe inserting the LEDs in deep nasal and toward cribriform plate and also sphenoidal sinus can work in terms of deep brain PBM therapy. And these are patents from the 2018, 2017, but the first idea came from 2004, I suppose, which was the pioneer and for this type of assumption. But apparently, I should say, at the MGH, Massachusetts General Hospital, researchers are working on the implantation of the LED, the probes next to the cribriform plates in order to radiation of the, orbitofrontal cortex in depression models. So that is a fascinating research that are conducting and are ongoing. So another approach, hypothetical approach, is the frontal sinus. That could be the doable option in terms of radiation of some of the prefrontal cortex in dementia or in bipolar or depression patients. Okay. And also, there are a couple of other types of radiation which we call indirect or abscopal photobiomodulation, which I can say which has been introduced by the researcher from Australia. And so the basic mechanism for these types of radiation is based on the systemic effects and systemic radiation of the, this circulation, immune system, stem cells, and so on. So in this method, the scientist or researcher just put the LEDs over the bone, long bone like the tibia, or even on the abdomen, and then after a period of treatment, a couple of positive beneficial effects on the brain could appear.
I'm going to point out, I want to just point out that we were, in talking, we're going to have these authors on, but also in talking with you and the researchers, remember that when we talk about transcranial photobiomodulation affecting brain, that could be called our direct effect. But there's as we spoke about, there's much tissue that has to be penetrated, it has to be affected in order for it to reach the brain. So each application of photobiomodulation will have a direct and an indirect effect, because you can't just say, I'm hitting one tissue with this light energy. So I think that's a very good point. That's a very nice slide that you made, how it has a systemic effect. Thank you.
Yes. You know what, actually, when we want to, transcranially radiate the brain, even in that case, we can put some of the indirect effects, like because we irradiate the scalp, which has some of the stem cells, or even subarachnoid, blood vessels. So the nitric oxide or other types of inflammatory factors could be affected in the scalp because of the stem cells, Right. So even in transcranial, we have the indirect effects. Okay. Other types of delivery approaches, which has been recently suggested by the Russian researcher, are intravascular laser acupuncture and whole body PBM, and plus intracerebral transcatheter PBM therapy, which all of these, not all of these, but intracerebral transcatheter method has been suggested by the Russian physician, I think the Maskovich, Mr. Maskovich, Dr. Maskovich. So that's a pretty amazing method for treatment in Alzheimer and Parkinson based on their 2019 remarkable study. And also intravascular is the same as the intranasal. Surprisingly, intranasal PBM has the same effects like the intravascular, but intravascular is kind of invasive. So And laser acupuncture is kind of another type, but specifically, radiation of the acupuncture point. And the whole body PBM, that's the amazing method for getting well-being and having the beneficial positive effects on the whole body, and plus brain can affect it positively from this method. Okay, let's talk a little bit in details about the penetration profiles and some of the transmission values for the light, how many photons can reach brain in the various types of species, and also human being had. So actually we have a literature review that has been published on the Photobiomodulation Photomedicine Laser Surgery journal in 2019. We showed that actually there's a reduction in transmission of the light, red and near-infrared light, because of the various amounts of thicknesses of the scalp and skull. So, and we also showed that, long story short, pig and mouse, biopsy mouse, have the approximately same laser transmission amounts and values for the, for the scalp compared to the human. So these two species can be, used in the laboratory studies in terms of, evaluation of the exact amount of light transmission. So we can see the values. I don't want to talk. Okay.
What you're saying is that the pig and the human, I see by your scale, have approximately, actually the pig has a 0.2% and to 10%. That's a little bit of a big range, but 0.11%. What are you trying to say? How do we look at this? What is a good model? How can they have similar penetration values of the pig and the mouse? I don't follow that.
The penetration depends on the absorption coefficient. That depends on the how many or amount of proteins, water, and the lipid, and etc. So in this laboratory and in this experimental models, we can offer, based on the literature, we can offer pig and mouse, the head components like the scalp and skull, as a very similar model to the human beings, head.
Very good. Because you can use
in terms of in terms of the transmission, not the scattering. Okay, in terms of this. Yes.
Okay, good.
Yeah, let's focus on the human brain. As you can see, the study from Jagdil et al. In 2012, they showed that the 633 nanometer LED lights can penetrate up to 6% over the right parietal, and the less penetration was achieved by 2% over the frontal. And for near-infrared, even then we can get 10% for the parietal. So these are from the LED lights, okay, and just the skull, okay. And for the brain tissue, laser light using 660 nanometer showed a kind of, similar. For the near-infrared light, we have the similar types of, similar values of penetration. But for the 660 red light, the hippocampus can absorb more light, and the transmission for the cortex is a little bit, more for this type of laser. Okay, the recent Monte Carlo simulation modeling for the 5-layer model of human head have shown that there are an exponential and also attenuation for the light transmission over the human head. And so 5.3% of the incident light over the skull can reach gray matter. And that's a little bit, inconsistent with some of those, experimental study that have reported their values. Okay.
So what do you mean by incident light? I mean, this is using light. What are you actually looking at when you did this? What kind of light were you looking at?
Okay. They use, as you can see here, they use 800 nanometer near-infrared light.
Okay.
And also based on the Beer-Lambert attenuation, light attenuation equation, how many photons at the first place in the incident light and how many inside the brain? Just only roughly 5%.
So 5% is I mean, you're going to kind of walk us through that, but we've seen 10%, right? 10% in some areas to reach the cortex, I think we were looking at. And then we looked at different layers. And now you're saying if we walk through this, we walk through this, this is the parietal cortex, and now we can have almost 5% that'll reach the gray matter. So very, very good. Thank you for that. Thank you for that.
My pleasure. Okay, another research even had fantastic results because some of the researchers have argued that why we use the pancranial or whole brain therapy instead of the single spot brain PBM. So this study and this diagram can show that when we irradiate the brain using the single spots like this 1, 3 over the frontal, parietal, blah blah, and then when we use the pancranial using 181 separately, even separated over the skull, the results of the retransmission will be changed. How? As you can see here for the single spots, at the depth of the 10 centimeter, okay, we will have 10 to the power of -8 of photons, incident photons in this case. But for pancranial approach, we will have the same value of the photons even in around the 7 centimeter. So we will have some, how can I say, intensify effect using the pancranial photobiomodulation based on this result.
This is great. Now, but could clarify me, I want to make sure I understand that. The depth of penetration for the pancranial that you have there in your red dot was about 7 centimeters, is what you said, correct?
Yes, sir, exactly.
But if I did a spot one, it went 10 centimeters, if I'm not mistaken.
10 centimeters. Almost is, the same for the all, spell over the skull, because it can circular is for the parietal.
Right.
If you can go the paper, you can find the details, but the values are, clear that pancranial has the beneficial effects because of the thwarting effects of the photons because we have the same phenomena in the radiation, the radiotherapy, because this is the electromagnetic, the photon doesn't matter in the invasive, spectrum or in the non-invasive. So because of the thwarting effects, this will happen.
So you're saying that can I just clarify? It seems that this is indicating that the single beam has a deeper penetration than the pancranial beam. Am I mistaken? Or you're saying that it's going to have, I'm just trying to figure out the normalized photon flux. So it seems like 100 is better than 70, but what you're indicating is that 70 is better than the other graph. You follow me?
Actually, inversely, as you mentioned, when we use the pancranial, we will have for example 10 photons at just 7 centimeter, but in terms of the single, we have the 10 photons in the 10 centimeter, which the pancranial is perfect because we will have the less attenuation until reaching the the 10 centimeter.
I'm not sure I understand that.
I mean, we will have the more photon in the upper depth.
Oh.
Yeah.
More photons in the upper depth, and that's
Yeah.
why is that
That's better?
Why is that better than the deeper one?
Because as you can see here, in the 10 centimeter, we will have even, for example 10 to the minus 9, but in this case 10 will have 10 to the minus 8.
Oh, okay, I got you now. It's gonna keep going. We just don't see it on the graph. Oh, wonderful. Yes, wonderful. Now that makes sense. Thank you, Farzad. Thank you, Farzad.
My pleasure. Okay, let's talk a little bit about the mechanism. What is a mechanism? As you know, in the photobiomodulation, we use very different the types of wavelengths depends on our photoacceptors. But in the brain PBM, because of the transmission matter and because of the scalp, the scientists just normally use the near-infrared light or far red near-infrared. As you can see here, 600 nanometer to 850 nanometer. So this type of light specifically is absorbed by mitochondria, and then the crucial and the main mechanism is the photo dissociation of the nitric oxide, which I will talk about this later. So and then afterwards we have the ATP synthesis and some of the modulation in ROS production, and keep going on the MMP, mitochondrial membrane potential will be changed, and some of the transcription factors like the NF-kappa beta and circular AMP will change after irradiation, or even at the time of irradiation. So for the light in the near-infrared region, 900 up to 1100 nanometer, we will have another types of photoacceptors that are located inside the ion channels. These are the heat light gated ion channels, mostly based on the calcium ion channels, which absorbing light can vibrate these channels and structures, and then the release of calcium can be occurred. Okay, and you can see here we have a wonderful mechanism for the light in the blue and green spectrum. As you can see, the photoacceptors are different for these two types. Cryptochrome's blue light receptor and green light sensitive opsin channels are dominant for this type of radiation. Let's keep going. As I mentioned, far red and near infrared wavelengths are commonly used, and as I mentioned, the main mechanism is based on the photodissociation of the nitric oxide from the central region of the CCO, or cytochrome C oxidase. As you can see, when light can reach to this part and then photodissociate of the nitric oxide, CCO can breathe better and mitochondria can breathe better, and then finally electron transport fastly and accelerate, and then we will have the boosting of the ATPs, and then eventually we will have the more ATP. Okay.
Wonderful graph. Wonderful graph.
Thank you. So neuroprotective benefits of brain PBM can be summarized in several types of, mechanism, as you can see in the level of cellular, neural mechanism. One of the most important mechanism is increasing of the cerebral blood flow, CBF. So, which the researcher believe that maybe increasing CBF is more than 70% of the whole, beneficial effects of light. And 30% depends on the long-lasting effects on the synaptogenesis, on the neuroplasticity, anti-inflammation effects, anti-apoptosis, increase of progenitor cells, and antioxidant effects. And most importantly, neuroprotective by the brain BDNF, GDNF, and NGF. So, the CBF is very important. And the early days of the brain PBM, scientists believing that the light can increase CBF via nitric oxide as a vasodilator.
Correct.
And this is the well-established, and well-accepted mechanism for the, PBM. Okay, let's talk about the neuropathological and in the preclinical study of the brain PBM. Based on our systematic review, and from 1976 to November 2019, okay, based on all of those papers that have been published on Web of Science and PubMed, there are 63 in vitro studies.
In vitro studies. In vitro
studies on the several types of cells, the primary neurons, astrocytes, and so on. And also neurotoxic models have been applied frequently. And actually, I can mention that the Wong Rayleigh, I don't know, I spell I don't know, maybe the Dr. Wong Rayleigh, I should check it. 2001, this is study. You can see one of the greatest study all the time of the brain, neural cell in vitro PBM, that they showed that cytochrome C oxidase, will increase, its activity will increase after PBM. That was the pioneering study. And after that, the researcher used the potassium cyanide and the MPP and etc. These models. So I will explain some of the main results and findings, which are today's clinical application actually are based on these, and these are very important. For example, one of the greatest studies all the time is the Huang study. Actually, this is from the book that has been published in 2019 in the Mike book, but the study was the 2011 Huang and Sharma's study. Dose response for the first time has been introduced for the neurons using PBM, as you can see, even for the ROS. I think it's for ROS generation, for the nitric oxide, and for the ATP. We have the same pattern of the change. That's fantastic. So they introduced 3 joule per centimeter square as the most effective dosage in vitro. So
go back one second. Go back 1 second, because that was an important.
Yes, sir.
Right down there, all of them are looking at 3 joules. They're looking at the different things. That's what you said. 3 joules per centimeter squared of an 810 nanometer laser, right?
Yes, sir.
Okay, so that's what you were looking at. And so what they did was they did different doses. They went up at different doses and saw of different, different joules and stuff like that. And they went up and saw the change. That's what you're trying to show on cortical neurons.
Yes, exactly.
So they
They
looked
were they were
at
look they were looking for
reaction oxygen species, nitric oxide release, and ATP activity. Very good.
yeah, they were looking for the optimum dosage for the neurons, because for the several types of body tissue and body cells, we have the another types of the dose response, and that was very important to they clarify that what is the best for the neurons.
Correct. More is not always better. Better is better.
Yes, exactly. Okay, and also for the MMP, also they showed that 3 joule per centimeter square can increase the MMP, I mean molecular membrane potential, as the same of the previous results. And also for the calcium releasing intracellular calcium, the story was the same, surprisingly. Okay, and that was a result from the intact neural cells, cortical neurons. In terms of the neurotoxicity, environment, another study, again, Huang et al., they showed that using the various types of toxics, like the COCl2, hydrogen peroxide, and retinol, they showed that photobiomodulation using 3 joule per centimeter square, based on the previous study, can decrease the ROS levels, the oxidative stress. So there are other results for the calcium also showed that intracellular calcium can decrease using the same joule per centimeter square, 3 joule per centimeter square. And also for the ROS, for the ROS in different types of toxics, they observed the same effects. So actually, they proved that 3 joule per centimeter square is perfect. Even we delivered that amount onto the cerebral cortex in human or even clinical, practice.
So just to give people a little bit of a touch here, neurotoxins are anything that insults neurons. So it doesn't mean that you have to take a curare dart or something, but anything that stresses a neuron is a neurotoxin. And what they're described here is that 3 joules per centimeter squared, or a certain dose of light energy reaching a nerve tissue, a nerve cell, makes it stronger. Correct?
Yes, sir,
More
exactly.
resistant to damage. That's a good thing. So we're not saying we necessarily, even though we demonstrated that blood flow is going to go up, angiogenesis is going to go up, brain is going to work better, synaptogenesis, everything like that. We're saying, okay, you take a brain cell, a nerve cell, and you put light on it, it's going to resist damage, correct? And this proved it. Excellent.
Exactly.
Excellent.
Yeah, as I mentioned, all of these are the fundamental, observed effects, which are, how can I say, based for the synaptogenesis, based for the neuroplasticity. So at the first place, even when upon the irradiation, nitric oxide can photodissociate, and then maybe after an hour or even less than 24 hours, we have the upregulation, downregulation of these, and maybe we will have some of the long-lasting effects like the synaptogenesis and neuroplasticity.
Wow.
Yeah. These are very, important.
Very good.
Okay, let's see what again, for the other types of, as you can see, neurotoxics, we will have the same results using ROS and NO, measurements. Let's keep going. And okay, one of the one of the greatest studies on the amyloid beta deposition and amyloid beta in vitro, model for the Alzheimer using photobiomodulation in 2019, which has been published in the Aging Journal, Aging Cell Journal. They clarify one of the greatest pathway, the activation of the PK a SIRT1 signaling pathway that has the role in the neuroprotection of the photobiomodulation this type. They suggested that at the first place, photobiomodulation can increase the activity of CCO, and then we will have the increase of the ATP production, and we'll have the more generation of the CAMP and PKN signaling pathway, SIRT1, and SIRT1 can inhibit some of the neuroprotection pathways based on the PGC1 alpha and RIRB8 of the pathway. So this is one of the fascinating, because when researchers use PBM in the animal models, for example, in amyloid beta Alzheimer disease model, one of the indication is what is the effect, what photobiomodulation can affect on the amyloid beta deposition. Apparently, this is one of the pathway and mechanism that can be explained for the amyloid beta, attenuation of the amyloid beta deposition using PBA. So another great mechanistic based article is that 633 nanometer light can promote BDNF gene expression via this pathway, calcium YARCA mediated CREB activation. So again, this is the very good pathway, because this has been put forward as a main signaling pathway in Alzheimer disease. So photobiomodulation can regulate calcium levels, and but their finding, their main finding was photobiomodulation can activate intracellular IP3 sensitive receptor stores, which induces an increased calcium. They were looking for that photobiomodulation, how can affect the calcium, the extracellular calcium or intracellular calcium that are stored in the IP3R, that they found that the IP3R stores are the answer,
And then they're released.
Release, yeah, exactly. And then the ERCA pathway will be, activated, and CREB, and which CREB can, be manifested in terms of neuroprotection, Okay. In terms of the in vivo study, animal studies, 145 studies have been published up to November 2019. So there's a gradual increase from the 2006, and so in various types of models, Alzheimer, neurodegenerative, depression, and also Parkinson, multiple sclerosis, and even addiction models, and as well as intact animals. So these are the types of studies using the non-contact transcranial radiation over the animal's head, using the contact manner, and also using the devices and restraint devices to, keeping, animals. Mm, okay.
Yeah, by the way, Professor Mitrofanis said that the mice really liked the red light therapy that they were doing on them. Somehow I don't know, as a researcher how you could say that, but he said that you turn off the light and they would look up at the red light, wanting more. So this is a great model to explain that there's different ways that they're accomplishing this act. So thank you for that.
Yes, sir. Yes, exactly. So a couple of, findings have been achieved by the various types of models, like the TBI model. It can protects the secondary brain injury induced by hypoxia, as you can see in the pathophysiological, sections. In terms of, combination therapy using metabolic modulators, the studies, some of the studies have shown positive effects. I want to just, quick moving on this. So spatial and episodic memories have been improved using the Alzheimer models, using the 810 nanometer for more than 5, separate papers. 810 was perfect for those in Alzheimer models. So it protects hippocampus from amyloid beta neural death using the antioxidant, is assessed by the antioxidant evaluations like the SOD, GPX, and etc. And in the animal, 80 models also can suppress the inflammatory factors like the interleukin 1 beta, interleukin 6, and TNF alpha levels, as you can see. And also, this is another great study that shows the implantable LED device for particularly mice. And this study is from the Lee's group.
These are surface or they're implanted? This is transcranial, so it's.
Yeah, this is the implanted mean. I mean this when animal moving in a state of awake. Yeah. And maybe when we implant the LEDs, it can they naturally irradiate the brain when they the animal
moving around, so it's really stuck on them. It's stuck on them. It's not inside their brain. I got you. Thank you.
Yeah. Yes. Exactly. So again, BDNF was increased using this method, and this is the long-term functional outcome.
So these have been against traumatic brain injury and the ischemic activities that it helps the brain. It keeps it healthy during insult. Okay, I like that.
Yes, exactly. And this is another fascinating study, John Mitrofanis group, that has been published in 2014, and which they introduced the way of implanting of the LED inside the brain on the substantia nigra, and the preliminary data was perfect, and they showed that the number of H plus cells were increased after laser photobiomodulation. So after 6 days, irradiation, and then in 2015, they started to investigation on the primates in the monkey, and the result was wonderful. And this has been published in Annals of Neurology, and one of the most cited papers on the in vivo, studies of brain PB, and this one is.
This is a great review.
Yes.
Professor Mitrofanis spoke about this, but I think that this is a great review because he showed the little optic fiber as it went in, you know, and now you give us a whole outline of it. And I think that the graphs, some things that he showed about is the movement. He was very, very excited about this monkey. And this was the one that he's showing here on the locomotive traces. This is the one when he said, this is how I know it worked. They called the monkey
Yes.
hero. He was the first monkey that got better, the first, you know, primate that showed to get better from this. So thank you for reviewing this for us.
Yeah, my pleasure. Okay, so in terms of the clinical studies that are very important, because we want to, you know, get and change the state from the preclinical to clinical. This is the all researchers in the field concern of the all researchers in the field. So 83 clinical papers have been published, and so as you can see from the 2003 or even 2000, then there's a, you know, a little, you know, gradual increase, but from the 2010 2011 there's a, you know, yeah, rapid increase,
exponential.
Yeah, exactly. So again, various types of, you know, conditions, neurodegenerative, traumatic, psychiatric, and also even in the healthy young and old individuals, which is pretty fascinating, you know, aspects of the PBM, which we can use for the normal condition, for the brain function in terms of boosting the sleep quality and the concentration, memory, learning capacity, and so on. So which I will talk about. Okay, in terms of the transcranial PBM for human cases, as I mentioned, a bunch of papers have been published, and as you can see, several types of tools and apparatus can be, you know, seen from this picture. Let's talk about one of the case report of the brain PBM for the dementia patients, which has been published in 2019. And in this paper, actually, we use 3 different PBM approaches: transcranial, intranasal, and body pad, or the patient's, you know, back. So we got that 4 weeks irradiation, 2 separate, using the all of 3, you know, these 3 devices, and combination with the red and near-infrared light, improve cognitive performance. This can be assessed by visual, spatial, executive function, orientation, or working memory. And surprisingly, we also showed that olfactory dysfunction was reversed. And that was a perfect outcome that we got. And the patient will tolerate it by the twice daily irradiation for 4 weeks. As you can see here, you can see some of the MoCA, Montreal Cognitive Assessment results that was significant after the 4 months. And this is another study using another types of headset, transcranial headset, which irradiate 4 separate regions over the head. I mean, over the DMN regions, default mode network. And this study has shown positive effects on the, functional connectivity within DMN network after 3 months of PBM in dementia patients. So in terms of the combination therapy for dementia patients, specifically Alzheimer's disease patients. This study have shown that 3 months of treatment using both 810 transcranial and intranasal photobiomodulation can increase cognitive performance and reduce some of the symptoms related to that, like the anger, wandering behavior, and also improve sleep in 5 patients. I mean 5 dementia patients, as you can see here. For the stroke clinical trials, there are 3 up to now. There are 3 different double-blind randomized clinical trials using 1,410 patients, which 808 nanometer laser was used, using it 1.2 joule per centimeter square. And the first one and the second one, I mean the studies and trials were successful, but the third one, a little bit, was disappointing. So maybe the dosage problem was the problem. So also the scalp thickness of the various types of peoples and patients, and also the onset of the stroke and the onset of the radiation, all of these made the results of the third study, disappointing. So as you can see here, the details are here. First one was from the 2009, 2007. Second one, 2009 and 2014. So, but there's no study for the stroke from that time,
After that time.
After that time, yeah. So in terms of the traumatic brain injury, whole brain therapy, brain therapy has been published in 2019, which all of these are very new, the clinical implications are very new in this field. So again, combination therapy using red and near-infrared can improve the neuropsychophysiologically scores, as can be, could be seen from the SPECT analysis that regional CBF can be, increased in 8 of 12 study of participants.
And Dr. Hipskin will be presenting also, as well as Dr. Lim. So we'll have a chance to look at this research in depth. And I think that this overview is just fascinating, sensational.
Yeah, let's go with the depression. The Schiffer study in 2009 was the first study on the neuropsychiatric application of PBM. So they use 810 nanometer LEDs, and this is the LED probe, and these are the near-infrared spectroscopy devices for evaluating the CBF change during and after the PBM therapy. They showed that 60% is on the depression rate and 70% on the anxiety rate can be achieved after PBM therapy, and also increased non-significantly cerebral blood flow during the light exposure. So, and because
what, son?
Because some of the reasons have been put forward in literature, because they used just the LED lights.
Go ahead.
And LED light, maybe some of the researcher believe that maybe LED light have the less penetration values compared to the laser light. Mm-hmm. And also maybe this is the non-significant, if you read their paper, they say this the level of the CBF was increased. You can see the increase, but it's not a, the
It
significant.
wasn't a huge one. So let me just point out that Dr. Schiffer is going to be on. And so this, again, I tell you, this review is excellent because it gives everybody a chance to see a taste of it before we dig into it deeper. But let me also point out, this is 2009. And I'm going to show you, if you look at that picture, that the place that they put the light, which is on the top corner of the forehead here, is far away from the emitter, the receptors. So
it's F3.
That, to see how far that is away, that you have, if they could pick up a signal this far away, 3-4 inches from where it was actually applied, it would be better if they had this applicator directly between the two.
Yes, sir.
So then they would really see it. So the point is, just to let everybody know, this is one single application showing a 60% reduction in anxiety and depression. That's
that was
pretty
impressive.
cool. In 2009. Yes. So I'm very excited to have him come on, and I see how you guys have gone forward with this neuropsychological and neuropsychiatric type of testing. But this is a great paper. Thank you for sharing.
Yes, sir. Yes, sir. And supporting your, claim, they just use the F3 and F4 points, and they detected FP1F and FP2 points. And so this is the why maybe they couldn't,
too far away. They reached the penumbra of the signal. They didn't measure the actual signal. So if they'd done the calculations based on you with a medical physicist, they could have done it much better.
Yes, exactly.
Thank you for this.
My pleasure. Okay, and this is the Sano's study. Has been published more recently, 2019, using 21 depressed patients using 823 nanometer LEDs and one session and much higher dosage. I mean, this is the scalp, 665 joule per centimeter square is the routine and commonly used. Even the Dr. Professor Gonzalez Lima, they used this type of, scalp joule fluence for the healthy. But for the depressed patients, Cassano used 60 up to 90 joule per centimeter square over the scalp using near infrared 823 nanometer, but Dr. Gonzalez Lima used 1064 nanometer, which has the kind of, different mechanism. It doesn't matter. The joule rate, the joule range is within the 60 up to 100 joule per centimeter.
Wonderful. And that's one of the things that you were talking about at the early slides. So it's very excellent that you bring it back up. Thank you.
Yeah. Again, the Hamilton Depression Rating Scale showed perfectly, improvement for, as you can see here, for the 11 patients that received the actual PBM compared to the sham group.
That's 11 sham and 10 active got the PBM of the near infrared light, dropped them down with their one session, and they followed them up for 8 weeks. That's phenomenal.
Yes, sir.
That's phenomenal.
Yes. And in terms of the Parkinson's disease, PD, you can find this the data on the book of the Mike Hamblin book, and so which I will show you the book if you want. That's a fascinating book actually. And so for the PD Parkinson's disease, they use different types of LEDs, and 670, 940 nanometer in one patient, 660 nanometer in one patient, 150 nanometer in one patient. But the overall was improved, as you can see in the handwriting, right? Handwriting of the patient has been improved after I think after
Well, they did that for months. They looked at
a
this for
month. Yeah.
months. So Mitrofanus
After,
touched on this, not in this
yes.
detail, and he gave us an overall view. But when they looked at the single patients, he showed this changes, and you're showing more changes here because that is the micrographia that
Yes.
they grew up more, their handwriting moved more, and their shaking, the ability of their hand to write shaped more. And there's different ways, but again, this is a pan-cranial approach with dual colors, two wavelengths, if you will. So that's thank you again, excellent overview of that really 2019, very fresh study that's still ongoing.
Yes.
And they're going to bring us more updates as we get the other speakers in are going to touch on this to a great extent. Thank you.
Yeah. You're welcome. So, as we are facing with Parkinson's disease, this is very important to reach photon into deep brain. I mean, the substantial network. So, what should we do is maybe the implanting of the LED or laser probe deep into the brain could be the great option, as those the scientists use for the deep brain DBS methods. So as you can compare these, if we want to radiate the brain of the mouse, we have a chance of the radiate the whole brain. But when we want to apply that for the human brain, we will have a problem. So we need to develop some of the techniques like this. So in normal human and healthy adults, a study from the Tian in 2016 has shown that right prefrontal cortex irradiation a little bit has the same results with the central frontal, transcranial radiation using 1064 nanometer diode laser, Nd:YAG laser. So this is one of the greatest studies that has been, derived from the Gonzalez Limas and Professor Gonzalez Lima and Professor Hanley Liu's, lab. So this was the origin of all of these studies that they work on the healthy, adults.
Wow.
I mean, the young and old adults. That the increase of the hemoglobin in the prefrontal cortex can, occur during the PBM therapy.
That's correct. So that's oxygen, blood oxygen proliferation.
Yes. So another study from the Wang, the same group, the Victor Wang, Dr. Victor Wang, so showed that the same laser in the same group of the healthy healthy individuals can desynchronize alpha and beta bands and subsequently stimulates ipsilateral frontoparietal occipital network and also contralateral parietal occipital network at the alpha band. And PBM-induced increase of alpha power is dose dependent. But how 77 up to 96 joule per centimeter square to skull, was the perfect, and as you can see here, they increase the alpha power.
Wow, this is a great the image of this, and we should have Dr. Wang on in a little bit. So just to let you know, this is the frequencies, right? The frequency band that you've written over there at the other side, the delta, theta, alpha, beta, gamma. And when you put this, this is the infrared, right? They like to use the infrared laser, the 1064 laser. It showed an increase in these bands as they're getting red in the minutes.
Actually, the most strong activation is found between 8 to 10 minutes after the onset of the radiation.
It's building up, and the areas that are getting the strongest were the alpha bands.
Also, even this effect can be, observed after the, completion of the irradiation, right?
Mm-hmm.
So that's very impressive for even for the, the alteration of the frequency bands and cerebral blood flow as well.
Wow.
So
Mm-hmm.
yeah.
And they did it on the right. They did it to the right.
I suppose, yeah.
I think that's what they're
Right.
saying.
Yeah, FP2 or the FP2.
Correct.
Okay, and what is the effective physical parameters for successful brain PBM? I'm going to, summarize this, and then we will have the conclusion part. Wavelengths is very important, okay? Large body of studies have employed specific wavelengths based on our literature review. And red to far red, 600 nanometer up to 670 nanometer. So helium neon laser also has been frequently used because of on the early days of the brain PBM, because this was the mostly employed laser. So near infrared region from 785 up to 850 nanometer are the dominant, wavelengths. I can say dominant. But 728 nanometer, known as the least effective wavelengths in terms of CCO absorption, cytochrome C oxidate absorption. So
we're saying you showed us the good ones based on the studies. These are the wavelengths that everybody used, and this is a great slide. You showed us the good ones, and at the bottom you showed us this is a bad one. So if somebody tries to sell you a device that says this 700 nanometer laser, or like this is that, don't believe them, right?
Yes. Based on, a couple of it, maybe the 5-6 papers that have shown the 730, 740, it's not effective, actually. And also you can find this claim from the speech of the Professor Mike Hamblin on the YouTube. Because I've learned that from YouTube, okay? And also based on the literature review. Okay, and so near infrared at 980 are using these days frequently on some of the studies, as you can see on the Professor Mitrofano's study on the PPD, Parkinson's disease. And we have 1064, the favorite wavelengths of the Texas University, the Professor Gonzalez-Limas group. So the mechanism for two of these, 980 nanometer and 1064, is based on the water molecule and calcium channels. And so, yeah, but if you want to know what laser and what LED and what wavelengths is better for brain PBM, you can see this figure from the Wang et al.'s study in 2018 that they reviewed that 810 nanometer laser, as you can see here, for more than 257 studies have used this wavelengths in the brain PBM because of the several reasons, like more penetration into the brain, having high scattering inside the tissue, and subsequently elimination of the large volume of the brain tissue, and highest absorption peak of the cytochrome C oxidate. This is the very important one. And also the absorption by ion channels. This 810 nanometer is, prominent, is prominent for use. So another parameter is fluence. The second important parameter is fluence. In vitro data from the neural cells have revealed a threshold for the stimulation of CC between 0.3 to 3 joule per centimeter squared. Okay. And average ranging from 0.1 to 15 joule per centimeter squared, found to to effectively, neural PBM in vitro and in vivo models. In human transcranial cases, fluence over the scalp ranged from 10 to 30 for neurological conditions, 30 to 84 for psychological or psychiatric conditions, and 15 to 60 for normal subjects. And overall, we can say clinical reports indicated cortical fluence approximately 1 to 2 joule per centimeter square, which are consistent with the 3 joule per centimeter square, approximately, is sufficient to achieve positive effects in clinical, setting.
So I wanted to thank you for this slide. This took a lot of work to put this together, and these are some of the things that you've been talking about for a long time. So to make this people aware of this, that in the studies that were done, there's these different fluencies for the different conditions. So you're still within the proper, everybody's got a certain range that you're sort of laying out for us. So I think that this is an excellent summary. Excellent summary.
Thank you. Actually, these are from the more than 400 different papers in the brain PBM.
Yep.
Yeah.
This is fantastic. Mm-hmm.
Thank you. The third parameter, I think maybe we can say the important parameter is operation, because of the different mechanism that the continuous wave light has and pulse light has. Studies have shown that the 1 to 3,000 hertz light, pulsing light, have been used over the more than 100 papers for this application in vivo and clinical. Because in the most of the in vitro cultural studies, they use just a continuous wave.
Constant wave.
Okay.
Wave, correct.
Yeah. When you look at the specific frequencies, you can find 10, 40, and 100, which are have the which has been proved that they have the significant beneficial effects more than or compared to other frequencies, especially 10 hertz. Okay, why? The superior safety and better biostimulatory effects of pulse wave mode in comparison to continuous wave mode is for some other reason. Because of the existence of on and off time, we don't have any, the increase of the temperature increase and the subsequent histopathological damage. Pulse wave mode has better penetration based on some of the, claim that has been put forward by the researcher, not the, the simulation modeling studies, but based on some of the practical and experimental, and mostly because of the experimental human studies, human head. So possible resonance can occur between different brain waves and the frequency of the light, laser LED lights and pulse lights. And also there is a resonance between the pulse duration of 100 millisecond equal to the 10 hertz and the half-life of opening and closing of the mitochondrial membrane ion channels. This is a very exciting.
Very exciting, very interesting work, and this work that we're going to see hopefully being expressed, that they're going to give us the sneak preview of this, is another operational variant. Thank you.
Yes. So as a conclusion, we can say that the neurotherapeutic effects of PBM using specific wavelengths of the red, near-infrared, and the far red, in those, as you can see, in a wide range of brain insults can work. Can have the beneficial effects. Models levels of red and near-infrared light 0.5 to 10 joule per centimeter square, show biostimulatory effects without any thermal damages or thermal effects. And PBM can increase CBF by releasing nitric oxide. And then we have the cerebral oxygenation. So PBM can reduce oxidative stress, neural cell death, apoptosis, neuroinflammation, and also a recent study can have shown that PBM can regulate the neurotransmitter, specifically serotonin and dopamine in the brain of the neuropsychiatric model, rat model. So PBM can even have the effects on those types of, the molecules inside the brain. So, and also PBM can have some of the long-lasting effects like the neuroplasticity, neurogenesis, and cytoplasticity, and most importantly, antioxidant, signaling pathway. So, development of new techniques like intracranial, intranasal, or via the oral cavity routes for effective light delivery to deeper structures should be done. So as a suggestion, I can say that the further studies are needed to reveal the optimum light parameters in vitro and in vivo models for evaluating the best pulse frequency, fluence, treatment session interpretation, and so on. So I mean, how can I say, for the treatment protocol should be, evaluated. The new trials for other psychiatric disorders, including schizophrenia, autism, bipolar, ADHD, and OCD will emerge in the future. Intracranial PBM, potential effect for those conditions that are associated with limbic system dysfunction, such as anhedonia, anxiety, and impaired emotional processing. And also for the Alzheimer's disease, it is possible that photobiomodulation can play even larger role in society in the upcoming years. Because of the ongoing and promising results that we have at the time. And combination therapy using pharmacotherapy and other, physical therapies like TMS, TDS, DBS, and electroshock therapy are recommended. And clinical or home-based PBM using laser LEDs will become one of the most promising strategies for rehabilitation in upcoming years. We hope so. So thank you so much. And
wonderful, Farzad. I want to thank you. I want to thank you for your expertise, all the work that you do, all the work that you've done, and your contribution to this field and your contribution to this summit. I really appreciate your time.
Thank you. Thank you so much. Thanks for having me.
You covered everything.